Good afternoon, students and viewers. Today we'll be discussing something about fermenters, bioreactors, and types of fermentations, which is a part of the Industrial Biotechnology paper BIT 4066 of Gauhati University. I am Dr. Hridip Kumar Sarma, Assistant Professor, Department of Biotechnology, Guwahati University. And as you all are witnessing, these two are photographs of fermentation and fermenters as such. Here what you see is two different types of fermenters with pH sensors. Sensors are very essential for any fermenter to work on. We will discuss about it a little bit later on. But before that, we need to know something about the basic design of a fermenter. When you look into this basic design, what you find is this aseptic inoculation pipe through which aseptic media is inoculated into this particular chamber, and you find a stirrer or a soft seal with a soft seal, and this basically rotates. This is because the stirrer helps in oxygenation as well as the mixing of different nutrients. This is the baffle, which basically it's a thermocouple kind of material which maintains the temperature. The sampling point through which you will be sampling the product. These are the impellers that helps in rotation and mixing of materials. The sterile air is very much required for aerobic cultivation of microorganisms, and this is the sparger, and this is the impeller shaft. So whatever you are observing here or witnessing is a diagram of a fermenter with one multi-bladed impeller. In batch fermentation, what we witness is generally a batch fermenter is used for fermentation process batch-wise, of cultivating a microorganism, a kind of microorganism batch-wise. And here you are observing a similar kind of thing like sterilizing steam inlet. The fermenter and culture medium is sterilized. Okay, before this is an example for penicillium production where the fungi is inoculated for growth of penicillium here in the medium, and the different nutrient inlet, the inoculation inlet, the thermocouple, which is nothing again I have already discussed here. It's nothing but the baffles are thermocouples that measures the temperature of a fermenting medium, and at the same time you can also maintain the temperature required for fermentation at a specific point. A cooling inlet is there, and here it basically cools if the temperature increases beyond a particular limit. Okay, so this is a water circulating kind of cooling inlet, and this water circulating inlet may be also connected to a water circulator. This is the motor that basically runs the pedal and mix the contents. The pH control is this. This is a probe that controls the pH and basically monitors that the pH is constant and doesn't change. There is no appreciable change in pH in the reservoir. This is a sterilizing steam outlet because heat will be generated during the process of fermentation, and that heat needs to be taken out. And the gas exhaust outlet is here to allow the gas. It may be carbon dioxide or other kind of gases that are being produced during fermentation. And this is the harvesting line, when you basically harvest the materials from below. So when we talk about the harvesting line, this is of great importance that the materials get deposited below. It is nothing but the product, and this is as a result of the fermentation process. So these initially are termed as products which come from the substrates which is there in the nutrients in the medium. So there are some of the examples of fermenters. This is the airlift fermenter, which is quite different from that of a stirred tank bioreactor. And here in this case, in airlift fermenter, what you see is air is sparged from below, and it keeps on circulating to the above and again below. So there is a circulatory mechanism out here in airlift fermenter. And this is a gas lift bioreactor. There is a gas flow outlet. These are bubbles. These bubbles are formed because of circulating media here inside the fermenter. There is the gas flow inlet. the gas distributor, because this allows gases, primarily oxygen or even fresh air, that can be added, because these kind of microorganisms that you go for fermenting in this kind of fermenter require a lot of amount of oxygen. They are basically meant for aerobic microorganism growth. And here the bioreactor types may be classified as continuous tire tank bioreactor, the bubble column bioreactor, air lift bioreactor, this is one of it, the fluidized bed bioreactor, the packed bed bioreactor, and photobioreactors. In case of if we want to cultivate algae, cyanobacteria, blue-green algae, and that kind of microorganisms that produce or that go through photosynthesis, you need photobioreactors. We'll go into solid state fermentation and we'll try to understand what is solid state fermentation. It's a biomolecule manufacturing process used in the food, pharmaceutical, cosmetic, fuel and textile industries, and these biomolecules are mostly metabolites generated by microorganisms on a solid support. So unlike that of the liquid media here, in case of solid state fermentation, we do not provide any liquid medium. We generally use a solid support to allow microorganisms to grow. This is best suited for the microbes that have the propensity to grow on a solid surface, those are dependent on anchorage and maybe filamentous in nature too. The process is, it consists of depositing a solid culture substrate such as rice or wheat bran on flat beds, and after seeding it with microorganisms, the substrate is left in a temperature controlled room for several days. This is quite similar to that of growing mushrooms on hay or stack of hay. In nature, filamentous fungi grow on the ground, decomposing vegetal compounds, and solid state fermentation enables the optimal development of filamentous fungi, allowing the mycelium to spread on the surface of solid compounds. And this is very much required if you want to grow filamentous fungi, primarily because if you go for a rotating kind of circulating type of bioreactors or fermenters, filaments cannot grow. Filaments can grow only under static conditions, and that's the reason why solid state fermentation is very good for fungal growth. The composition of growth medium guides the microorganism's metabolism towards the production of enzymes that release bioavailable single molecules such as sugars or amino acids by carving out macromolecules. Now, these enzymes are being produced continuously by the growing mycelia of the fungi, and from where you can harvest later on the enzymes from the substratum or the substrate being used. Therefore, when selecting the components of growth medium, it is possible to guide the cells towards production of desired metabolite or metabolites, mainly enzymes that transform polymers like cellulose, hemicellulose, pectins, proteins into single moieties in a very efficient and cost-effective manner. And you can cultivate microbes on heterogeneous solid substrates that requires expertise to maintain optimal growth conditions. So this is one of the biggest advantages of solid state fermentation, that you can cultivate microbes on heterogeneous solid surfaces, because you can use different kinds of substrates to grow a particular mycelium. What is more important out here is to attain a maximum mycelial growth during fermentation. Airflow here is monitored, and it is key because it impacts temperature, oxygen supply and moisture. These things are key to any kind of fermentation process. Temperature, number one. Number two is oxygen supply, and moisture, of course, is a much needed material, or I should say that much needed a kind of most important part to find out whether or not these fungi are growing. In order to maintain sufficient moisture content for the growth of filamentous fungi, waterlogged air is used, because fungi primarily grow under hot and humid conditions. So you need to maintain those things. Solid state fermentation does not require a completely sterile environment, as initial sterilization is associated with the rapid colonization of the substrate by the microorganism. This is also an important factor because fungi grow very fast. Their doubling time is quite faster. They can overcome the growth of other microorganisms, so you need not go for too much of sterilization, which is not true in case of many other microorganisms that also support the growth of important metabolites. Uses and applications. Traditionally, solid state fermentation has been used in Asian countries to produce koji. In Japan, they produce koji using rice to manufacture alcoholic beverages such as sake, or koji using soybean seeds, and even sausages, soya sauce, and others. In Western countries, the traditional manufacturing process of many foods uses SSF. Examples include fermented bakery products such as bread, or for maturing of cheese. So this has been in place for hundreds of years, but now scientists and industrial producers, industrially the kind of industries which are involved in making bakery products, food products, they are utilizing this facility of solid state fermentation. It is well suited for the production of various enzymatic complexes like multiple enzymes. Examples are pectinases, hemicellulases, cellulases. So they are produced in a lot more amount, and this is quite advantageous to many other processes. So if we look at the advantages, the process is very simple, it is cost effective, and less effluent released, and it reduces pollution, and these are some of the other advantages that are being mentioned starting from here. And if you look at the process optimization, includes the optimization of physicochemical and biochemical parameters, initial moisture, pH, pre-treatment of substrate, maintaining relative humidity, and the temperature of incubation, agitation, aeration. All these things can also be optimized. And these are some of the substrates being used in solid state fermentation, like sugarcane bagasse, tea waste, wheat bran, sawdust, apple pomace, coconut oil cake. And these are two examples, as you are witnessing out here, is the growth of fungi like trichoderma on wheat straw, and from where you can extract out the biomass and then finally go for purification of enzymes and that kind of things. So this is a flow chart of the process that is widely used in solid state fermentation. I would advise the students to kindly go through the flow chart and understand themselves. Here I am also giving some other example. I am trying to provide some example of solid state fermentation, the products that come out from solid state fermentation. For example, mushroom products, soy sauce, cheese, tempeh, leaching of metals, organic acids. So you see, you can also leach metals using this kind of fermentation process. And this is how you can grow cellulase. Yeast strains can be included in some kind of sludge, and then you can produce cellulase. You can extract out the cellulase. It may also produce ethanol, so you will also get a fuel. Ethanol is a source of fuel widely used these days. So this way you can also get multiple products out of solid state fermentation. Now this is the design of a solid state fermenter. Here it's a kind of drum-like material, and from below there is forced air supply. So this is quite required, as I have told you, that without air you don't see growth of microbes. Air is required, oxygen is required, and at the same time you need water, because the humidity has to be maintained, and under such conditions this water is being sprayed. This is water, this is spray nozzle, okay, the liquid supply. And here you find in this paddle type mixing device, where maybe you will be keeping your substrate on the surface, and you will be putting your yeast or any other material, the microorganisms, on the top. So it will be sprayed by this water. The humidity chamber will be, this acts just like a humidity chamber, and this will ensure that the substrate is being converted into product, or else the materials that you want and can be recovered for final products through this place, through this nozzle out here. This is the exhaust line and evaporation chamber. So this is a typical diagram of a solid state fermenter, which is widely used nowadays. This is a layered kind of state fermenter, where you will find that this is the culture substrate, where you have the culture of the microorganism layered on the top. Just like the previous one, I had showed that here this is basically the cultured substrate where you can layer the culture, as well as you can sprinkle the seeds of that particular bacterium or yeast or fungi on the top of it. Primarily fungi is used for this kind of fermentation, as I have noted it earlier. These are module bases, these are cooling devices. Cooling devices are primarily used because during fermentation a lot of heat is produced. So too much of heat is deleterious for the production of enzymes and other materials. And this is the air inlet, this is the water supply, this is water discharge, provided if there is a lot of water that gets accumulated out here, has to be discharged. Otherwise it will also increase the water content, and that is not good for the health of the growing fungi. Here is the components of a typical laboratory scale bioreactor. Now here we are discussing about a laboratory scale bioreactor. I am trying to compare a particular solid state fermentation unit to that of a laboratory fermenter, and here we observe the materials are being quite expensive. Like for example, this is a liquid medium. You have an anti-foam port. This is very much required, important. Let me tell you that during the fermentation process, carbon dioxide is produced, and that basically produces a lot of foam. Okay, and that is not good for the healthy growth of the microorganism. Too much of foam will reduce the growth efficiency, and therefore you need to add sometimes anti-foaming reagent. There is the steam, the nutrient and or the inoculum is inoculated through this port. There is a sterile nutrient medium as referred earlier. These are the impellers that will stir the medium and will ensure that there is a homogeneous mixture of materials, and there is a homogeneous growth of the microorganisms also, and that the microorganisms need not aggregate and accumulate together. So this is the reason why you need impellers. And this is the air acid base balance system. There is a pressure gauge which measures how much pressure is there. Additional pressure beyond a certain limit is allowed to go out, and this is the filtered waste gases. Carbon dioxide or other kind of gases that may accumulate during the process of fermentation is allowed to go out through these filtered waste gases. The cold water outlet, this is nothing but this entire material is jacketed. If the temperature increases beyond a certain limit, then this particular cold water outlet is basically this movement of cold water ensures a homogeneous temperature gradient in the entire fermenter. It's a cooling jacket, and there is the steam, the harvest pipe, the sparger. The sparger basically ensures that there is a homogeneous distribution of air into the medium. This is from below. Sometimes you find some of the spargers from above. So the bioreactors depend on the kind of microorganism that you need to grow. Now we'll discuss something about the submerged fermentation. It's a method of manufacturing biomolecules in which enzymes and other reactive compounds are submerged in a liquid such as alcohol, oil, or a nutrient broth. The process is widely used for a variety of purposes, mostly in industrial manufacturing. So these are the differences between the solid state and submerged fermentation. I would like to ask the readers and the students to kindly note down the differences between solid state and submerged fermentation. These things are clear out here. This will let you understand more efficiently about the differences between submerged and solid state fermentation. So basically, submerged fermentation is a commonly employed technique that is widely used for citric acid production by some microorganisms. And the two types of fermenters, the stirred tank fermenter and tower fermenters, are widely used for submerged fermentation process. So this is a stirred tank fermenter and a typical submerged tower fermenter for vinegar production. So it's not only about citric acid, vinegar is also produced by a typical submerged tower fermenter. I would request the readers to go through the different parts of this fermenter types, like the baffle, the jacket, the impellers, types of impellers used, what are the different kinds of impellers, what's the need of the impeller, and what's the importance of acid base balance anti-foam reagent, how does it help in maintaining the growth of microorganisms if you decrease the if you decrease the foam, the air compressor, steam, water return, all these things are out here. How do you measure the pH, how do you monitor the pH, the dissolved oxygen, how is it important to the growth of the microorganism, the temperature, how it has to be maintained. So these are important attributes of fermentation. What are they? They are the pH, dissolved oxygen, temperature, acid base balance, foam, formation of foam and use of anti-foam reagent, exhaust air, and the kind of air that is let in, and the amount of sterilized medium that is let in. So this is basically a typical batch fermenter, whereby batch by batch you produce the products made by a particular bacterium, and you take out the materials to harvest the products, and then you again put new fresh medium, sterile medium back into the fermenter. This is batch by batch you prepare a kind of fermentation, and that is why it's called batch fermenter. So this is all about the submerged culture process. You can go through and understand yourself how is the submerged culture process important in submerged fermentation. And this is an example of fermentation of citric acid production. I would request and advise the students to go through this, the biochemical basis of production of citric acid. So the importance of production of citric acid is an intermediate. As you all know that citric acid is an intermediate of the Krebs cycle. So the acid can be accumulated by using one of the following: by mutating the organism that you are utilizing for fermentation. You mutate it to such an extent that it does not go beyond a production of citric acid. So this may be by mutating some of the metabolic pathways or regulatory mutants, that is used as a mutant lacking in enzymes of the cycle. So if there is no more enzyme that can convert citric acid to the next intermediate, then automatically you find that citric acid is the ultimate product of that particular cycle. So that is how you can use mutant organisms. You can inhibit the free flow of cycle through altering the environmental conditions like temperature, pH, medium. So if you alter any of these, you may find that beyond citric acid you don't find any other intermediate being produced. Uses in the food industry, used in medicine and pharmacy. I can go through the uses of citric acid from here. So these are examples of, again, citric acid production through airlift bioreactor. Airlift bioreactors are similar to bubble column bioreactors, but different in the fact that they contain a draft tube. It's always inner tube. So basically when we call a draft tube, this is a draft tube. Inside that of a central tube you find another tube. So this tube is hollow at both the ends, which basically allows the movement of molecules, the cells in and out of the tube. This keeps on a circulatory motion, and a lot of air is inlet as gas is let inside. So this air bubbles alleviate the process of circulation. There's an external loop which improves circulation and oxygen transfer and equalizes the shearing forces in the reactor. So this is an important part of an airlift bioreactor. When we talk about surface fermentation, and this means the fermentation takes part on the surface of the media. Okay. And there are a lot of advantages and disadvantages. You can go through as to when do we need to go for surface fermentation. Ethanol conversion into acetic acid, the rest of the substrate is used as biomass production or lost by volatilization. So ethanol conversion into acetic acid is basically a surface fermentation phenomena. These are the differences between submerged and surface fermentation. I would like to request the readers and the students to go through and understand the differences between them. So here, this is the best example. You look at lager yeast, the chills down here, so they get deposited at the bottom, while ale yeast hangs up on the top. So this ale yeast is basically a surface fermenter, while lager yeast is a bottom fermenter. So two different types of yeast, but they are used for preparing different kinds of beers. Now you are also observing, witnessing out here is that they need different types of temperatures for optimal growth. So that is why, dependent on the kind of microorganism or the type of fermentation they go for, you can choose and you can optimize the fermentation parameters. We'll now next go to process control and optimization of fermentation parameters. And amongst which the most important is the microbial growth kinetics. Let me tell you that fermentations may be carried out as batch, continuous or fed batch processes, and the mode of operation to a large extent is dictated by the type of product being produced. So what product you want, what kind of product you are going for, that will depend on what kind of fermentation you need to go for, like batch, continuous or fed batch. Now typically in batch fermentation, we'll discuss something about batch fermenters. In batch fermentation, typically you will find the growth of a microorganism following this sigmoidal curve. The growth phase is also referred to as this portion is also known as the log or exponential phase, and from where you will find that after a particular time the entire nutrients will be utilized by the microorganisms and there will be a stationary phase, there will be no growth. But here at this stage, all the microbes will be attaining a maturation stage and they will be producing the metabolites that are of use to us. And finally, this will be followed by a death phase where the microorganisms will die off. And here below is the lag phase, which is nothing but the starting phase, the kind of amount of time being required to start off the fermentation process. So this is atypical of any bacterial culture, which we have learned about microbial growth patterns earlier in microbiology. So batch culture is a closed culture system that contains an initial limited amount of nutrient, and the inoculated culture will pass through a number of phases as has been discussed here. Now in the lag phase, no growth takes place and hence is considered as time of bacterial adaptation. So this is the time of bacterial adaptation when the bacteria will try, or the microorganism will try to adapt itself to the environmental conditions inside the fermenter. And after that, there will be a burst of increase of respiration, there will be a burst of increase of metabolic activity, and therefore you'll find huge growth pattern, which is nothing but the log phase, log or exponential phase. For commercial purpose, the length of lag phase should be reduced as much as possible, and this is done by using selective inoculum, because the longer is the lag phase, it will take more time for the bacteria to adapt to the environmental conditions. So for commercial purpose, basically the industrial purpose, it is desirable that the lag phase should be short enough so that you can get the product much more faster and earlier. Now during exponential phase, the growth of cells gradually increases and obtain a maximum rate, and the kinetics of this exponential phase may be described as ddx of x equal to mu x. So this is the equation which basically describes the kinetics of the exponential growth of a microorganism in culture. Here x stands for concentration of microbial biomass, and mu is the specific growth rate of the microbe in hours. So what you need to do, if you integrate both sides, ddx of x equal to mu x, if you integrate both sides, you come to an equation which is xt equal to x naught e to the power mu t. This equation very much important because this basically suggests certain parameters like original biomass xt equal to x naught e to the power mu t, where x naught is the original biomass concentration, xt is the biomass concentration after time t, and e is the base of natural logarithm. So on taking again natural logarithm both sides, if you take ln xt by ln of x naught e to the power mu t, now since in logarithm anything to the base e equal to 1, you will be getting ln of xt equal ln of x naught plus mu t, which is an important equation here, and this equation is similar to that of the equation of a straight line y equal to mx plus c. This is y equal to mx, this is mx plus c. So y equal to ln xt, mx equal to mu t. You can just consider this as the equation of a straight line, and thus plot of natural logarithm of biomass concentration against time basically yields a straight line, the slope of which is equal to mu. So mu is nothing again but the specific growth rate per hour of the microorganism. During exponential phase of growth, nutrients are in excess and the organism grows at its maximum growth rate mu max for the generalized conditions. It is very easy to visualize the exponential growth of single-celled organisms that replicate by binary fission, for example, yeast cells, animal and plant cells. But it is very difficult to visualize growth of mycelial organisms, which show apical growth exponentially. Therefore, this kind of equation doesn't hold good for filamentous microorganisms like fungi. This is good for only single-celled ones that do not accumulate together. In this, under such circumstances, you can easily consider or you can easily determine the plot of natural logarithm of biomass concentration. Plomey in 1959 was the first to suggest that filamentous fungi have a growth unit which is replicated at a constant rate. And this was again supported by Trincy in 1974, who demonstrated that the total hyphal length of a mycelium and the number of tips increases exponentially at approximately the same rate in fungi. So when a fungi basically grows on a substrate, what really happens is that its tip starts increasing, and the tips can grow from anywhere or any branch. So this number of tips now grows exponentially at approximately the same rate of the fungi. So this was demonstrated by Trincy in 1974. Therefore, we can say that the volume of hyphal growth unit exceeds a critical volume, which initiates a new growing point for another branch filament. And this initiation of new branch point is equivalent to the division of a new cell when it reaches that critical volume. So, therefore, what we understand from these suggestions is this, that Plomey and Trincy wanted us to understand that basically this filamentous fungi, the growth of filamentous fungi is basically dependent on critical volume. And this critical volume helps to determine the division of a particular new cell and the branch points. The specific growth rate of a filamentous fungi can thus be calculated as ddt of x equal to mu x. Here in this case, in case of filamentous fungi, x is replaced by h or a, where h is total hyphal length or number of growing tips. Any one can be calculated. Henceforth, specific growth rate in such case is dictated by increase in hyphal mass. In case of filamentous bacteria, specific growth rate is determined by increase in hyphal mass, total length or number of growing tips. These three are the important conditions to understand the specific growth rate in fungi, and these are attributes. In submerged culture, sac, flask or fermenters, a mycelial organism may grow as dispersed hyphal fragments or as pellets. The growth of these pellets will be exponential until the density of pellets results in a condition called diffusion limitation. So, as mentioned earlier, we said that it is wise or it is customary to allow growing fungi on solid state conditions, because then the ramification of fungal filaments is much more profuse. It grows much more at a higher pace, at a higher rate, and you can get the materials of your need at a higher concentration. But if you grow this kind of filaments in sac, flask or fermenters, you are not allowing them because they will be on rotation and movement, so they cannot have a support, and as such there will be one condition which is called diffusion limitation, which will not allow them to grow at a pace that is required for getting the products. In diffusion limitation, the central biomass of the pellet will neither receive a supply of nutrients nor excrete toxic secondary metabolites in culture. Therefore, the actively growing zone for new filament is the outer cell of biomass, which can be expressed as below. And this has been suggested as such. You can go through this equation, which is root over mt equal to kt plus root over m0 divided by s, where M0 is the mycelial mass at time 0, Mt is mycelial mass at time t, Kt is diffusion constant at time t. So this equation was given by Pritt in 1975, and a plot of cube root of mycelial mass against time will yield a straight line, the slope of which is equal to K. Growth results in the consumption of nutrients and excretion of products and metabolites. After a certain time, the growth of culture decreases and growth ceases. So the cessation of growth is primarily due to depletion of some essential nutrients in the medium, because we added a particular substrate at a particular concentration. Once the concentration decreases, the depletion of essential nutrients is also obvious. B is accumulation of autotoxic products of the organism in the medium. So if there is overproduction of toxic metabolites in the medium, the microorganism will not grow. And a combination of both can also cease the growth of fungi under such conditions. Now, the nature of limitation of growth of a microbe may be explored by growing the organism in the absence of range of substrate concentrations, like carbon and nitrogen sources at different molar or millimolar concentrations, and plotting the biomass concentration at stationary phase against initial substrate concentration. So here what you are witnessing is a graph, and here we are trying to show you the effect of initial substrate concentration on biomass concentration at stationary phase in batch culture. Here A to B, there is a burst of biomass production. From B, mid of B up to C, you find a kind of stationary phase, and from C to D is complete stationary phase. There is no more formation of cells. The biomass production is over, and here there is accumulation of metabolites. Here A to B means an increase in initial substrate concentration that yields a proportional increase in biomass production at stationary phase. This is primarily this portion from A to B in case of filamentous fungi, or is described by the equation x equal to y SR minus S, where x is concentration of biomass produced, y is the yield factor, the gram of biomass produced per gram of substrate consumed, SR is the initial substrate concentration, and small s is residual substrate concentration. From the above plot, it becomes evident that from zone A to B, the residual substrate concentration, here residual substrate concentration S, small s equal to 0 at the time of cessation of growth. Meanwhile, in the zone C to D, any increase in initial substrate concentration does not give a proportional increase in biomass. This may be due to exhaustion of substrate or accumulation of products. In the transition zone B to C, the utilization of substrate is deleteriously affected by the accumulation of toxins. So here between C and D, this is because of accumulation of toxins, and which are nothing but the secondary metabolites being produced by the microorganism in culture during their growth, and this attains to such an extent that this does not allow the further growth of the microorganism. At the same time, there is nutrient limitation, because the amount of nutrient initially added for the growth of in the growth medium for the growth of the microorganisms has been exhausted during the growth from phase A to phase B. This is basically the entire fermentation process. So this equation holds good if you want to calculate the yield based on concentration of biomass produced, based on the initial substrate concentration, and at the same time the residual substrate concentration. In the next class, we will discuss more about what is residual substrate concentration, how to determine residual substrate concentration, and how to calculate the cessation of growth based on yield. Thank you once again for your kind attention to this particular presentation.